A wave of new technologies is sweeping across biology. Flow cytometry can now read dozens of markers simultaneously from hundreds of thousands of cells every few minutes. Functional MRI can deliver a movie of the brain at work. We can monitor the activity levels of tens of thousands of genes and score millions of DNA variation sites in an individual. For less than $10,000, we can sequence entire human genomes, and $100 genomes are already in sight. Plummeting costs mean that within a year, individual labs all around the world will be gathering terabytes or even petabytes of data. These data are already being linked to an array of other information to offer a fundamental new understanding of living systems. We have crossed a threshold into the realm of the new biology.
I don't know whether the correct term is the new biology or the golden age of biology. I just know that the level of enthusiasm that I see right now in the community and the way it is coming together through large integrative processes is unprecedented.
We are at the beginning of a new era. We call it a new paradigm.
The new biology, as it's evolving today, is basically many different fields realizing that they can collect a lot of information. They can bring to bear computational tools to mine the data that can give us results that are correlating to both biological and clinical outcomes. The goal of any kind of research is not just to create information, but to actually distill it to some level of wisdom. That's the goal. Data, information, knowledge, wisdom.
I think the new developments in ultra-fast sequencing will take us into a new era again in biology.
The new biology will transform every aspect of human health, from food production to cancer treatment to disease surveillance. And this scientific revolution has one organizing theme: assembling complex, high-dimensional bits of data into a realistic portrait of life.
There's a big realization now that actually we have to put it all back together again to really understand the intricacies of the very complex systems that we're looking at. That's been really difficult to do, but now with new technologies, new understanding, which is very much founded on our previous understanding, we're beginning to be able to do that. So it's an incredibly exciting time.
The new biology is systems biology realized. And it's realized by the new technologies that enable us to take the comprehensive snapshots of the system, but it's also enabled by the new ways of thinking, of integrating information, building complex models. The human mind is an amazing engine for putting information together and understanding it, but it has to be presented with the information in the right kind of way.
Think about how your mind organizes data, sound, pictures, and text, to let you grasp a story and feel its emotional impact as you watch this Google ad.
Hello. Bonjour.
Très très mignon. Allô?
Le commandant et toute son équipe espèrent que vous avez passé un agréable.
Each movie is a frame of millions of pixels, and then you have thousands of frames through the course of a movie.
If you looked at the ad the way that old biology looked at living systems, you'd see each pixel as an average value over time.
Say, okay, understand this movie from this average of all the pixels. You'd never achieve understanding that way.
Or you'd look at one raster line and see how the pixels' intensities change over time. It's more information, but meaning is still far away. Now, add a bit more data, fragments of frames, a word here, a symbol there. Next, add context. The words are appearing on a screen. Whose computer is it? More context. Someone is performing a series of searches, asking questions. Now you see a bit more, but not in the right order, and you can't tell what's most important. Ideas are flying in your mind, but they don't add up to the story the ad intended to tell. Now add information about which words are most important, the ones in the query box. But still, you lack the right order to get the truth of the story. You might come up with something that seems sensible, but is wrong. Someone is planning a vacation in France or looking for a job with an airline. If you add sequence information, you'd get a partially true story. But with information gaps, you can miss the whole point of the ad. It's a love story. Now, bring in a new layer of information, sound.
Allô?
And at last, the entire true story is revealed with full emotional impact that completes our understanding. Data, information, knowledge, and knowledge about that knowledge. That's what you need to achieve wisdom. Can we look at living systems in the same big picture way we watch a movie? The old way of doing biology was a lot like looking at a few pixels averaged over time. You'd study a handful of proteins, and on a good day, you'd end up with a simple linear pathway that explained a few of the steps that cause disease phenotypes.
There is nothing in biology so complicated that if viewed in the right way does not become still more complicated. We know a tiny fraction of 1% of all there is to know.
I think the real promise of molecular biology was, for the first time ever, we could look at the individual components, that is, the individual genes that encoded the information for making organisms. And I think in a naive reductionistic view, people thought by understanding the individual components, they could come to understand the collective behavior of those components. And that turned out to not be true at all.
The new biology is corralling data into meaning. The data are large numbers of observations of molecules over time and individuals, and at a hierarchy of levels. Sophisticated algorithms can reconstruct causal molecular networks that represent the physiological states associated with the complex traits like disease. These networks aren't islands. They are highly interconnected in a complex system. The intricate multi-scale network of life can be visualized in this model. Each network, defining physiological states of the system, maps to a region. When one network affects another, that's shown by a directed arc. Interacting networks are often found within a single organ or tissue in the body. What happens when you look at the interactions of networks across tissues? These are shown by links inside the model. It's only become clear in the last few years that the interactions across tissues may far outnumber those within. This is a compact representation of a system-wide molecular network that underlies the physiological states of an organism. It enables higher-order predictions of how an internal or environmental perturbation will ripple through a living system. This link between molecular biology and physiology is the central insight of the new biology. What's making the new biology possible are novel bioimaging technologies. Functional MRI, for example, shows brain activity over time. It's become a key tool of cognitive science.
At MIT, one of the cool things that we've discovered using functional MRI is that there are at least a few distinct parts of the brain that conduct remarkably specific functions. One of the discoveries is that there's a region in the right hemisphere that's specialized not just for simple perceptual operations, but this one is specialized for thinking about what other people are thinking.
The fMRI system delivers brain data in the form of activity levels over time of each of tens of thousands of voxels, or 3D pixels. A voxel usually represents around 1 million neurons.
In a typical MRI scan, we scan subjects in there for about 2 hours. We collect about 3 gigabytes of data. My lab collects about 10 terabytes of data a year.
At the level of individual cells, another high-throughput imaging technology is flow cytometry. It can now deliver a gigabyte per minute of high-dimensional data. And DNA sequencing technology has come a long way since its birth.
We've gone from the early machine we developed in the mid-'80s to machines now that are 20,000 times more rapid, less expensive.
Second-generation machines have high throughput, enabling broad-scale human sequencing for the first time. And third-generation technologies are on the way.
The single molecule real-time paradigm allows you to see the individual components, the characters, if you will, in the play of life, in their appropriate context, but one at a time.
This smart approach eavesdrops on nature's own gene readers, polymerase enzymes, to get DNA and RNA sequences. The same approach can also examine proteins.
In the near future, we will have a technology which could sequence a human genome in $100. But when you have these $100 genomes, how you could read it, how you could understand it, how you can use it.
One effort to exploit massive amounts of genetic data is taking place at the University of Tokyo. Biobank Japan has data representing the DNA of 300,000 patients spanning nearly 50 diseases. Yusakai Nakamura is the founder of the project.
それらの病気の方の DNA あるいは遺伝子多形を比較することによって病気になりやすさを見つけより良い治療法を患者さんに提供しようとしています。医療の体系そのものがこのバイオバンクジャパンを通した膨大なその臨床情報からゲノムの情報まで合わせた解析によって変わってくるというふうに考えています。
One more technological advance is needed if the new torrent of data is to be harnessed. A leap in computing power.
We're generating more data than you could ever go back into the lab and validate.
There's an intangible cost with the difficulty in managing this glut of data. It's a logistical nightmare.
その理由は、そのデータの取得が自動化されてきたことなんですね。だから大量のデータが出たと。そうすると大量のデータは、自分の鉛筆と紙だけでは計算できませんから。
We used to think terabytes of data was big, but that's nothing these days. Now we're into peta or even exabyte scales of data.
We now have seas of data which are drowning us. People now are coming together, realizing that the key to mining data is computation. Look at Google, look at Amazon. These are key industries that have realized that there's an awful lot of data that can be measured from the herd, from crowd effects,